Hello everyone. Welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of the ribosomes and the process of protein synthesis. This module would help us to gain a pretty good idea about the structural and functional organization of ribosomes and the distinguishing features between prokaryotic and eukaryotic ribosomal protein synthesis. Structure and function of ribosomes. Eukaryotic cells are distinguished from prokaryotic cells by the presence of membrane enclosed organelles within their cytoplasm. These organelles provide discrete compartments in which specific cellular activities take place, and the resulting subdivision of the cytoplasm allows eukaryotic cells to function efficiently in spite of their large size. Ultrastructure of ribosome. Ribosomes are sites of protein synthesis in both prokaryotic and eukaryotic cells. Ribosomes are usually designated according to their rates of sedimentation, 70S for bacterial ribosomes and 80S for the somewhat larger ribosomes of eukaryotic cells. Both prokaryotic and eukaryotic ribosomes are composed of two distinct subunits, each containing characteristic proteins and ribosomal RNAs. The fact that cells typically contain many ribosomes reflects the central importance of protein synthesis in cell metabolism. E. coli, for example, contain about 20,000 ribosomes, which account for approximately 25% of the dry weight of the cell, and rapidly growing mammalian cells contain about 10 million ribosomes. The structure of prokaryotic and eukaryotic ribosomes is similar, although they differ in some details. The small subunit designated 30S of E. coli ribosomes consists of the 16S ribosomal RNA and 21 proteins. The large subunit 50S is composed of the 23S and 5S rRNAs and 34 proteins. Each ribosome contains one copy of the ribosomal RNAs and one copy of each of the ribosomal proteins, with one exception. One protein of the 50S subunit is present in 4 copies. The subunits of eukaryotic ribosomes are larger and contain more proteins than their prokaryotic counterparts have. The small subunit of eukaryotic ribosomes is composed of the 18S rRNA and approximately 30 proteins. The large subunit 60S contains the 28S, 5.8S, 5S rRNA and about 45 proteins. Like tRNAs, rRNAs form characteristic secondary structures by complementary base pairing. In association with ribosomal proteins, the rRNAs fold further into distinct three-dimensional structures. Functions of ribosomes. Ribosomes are the workhorses of protein synthesis, the process of translating mRNA into protein. The mRNA comprises a series of codons that dictate to the ribosome the sequence of the amino acids needed to make the protein. Using the mRNA as a template, the ribosome traverses each codon of the mRNA, pairing it with the appropriate amino acid provided by an amino acyl tRNA. Amino acyl tRNA contains a complementary anticodon on one end and the appropriate amino acid on the other. The small ribosomal subunit, typically bound to an amino acyl tRNA containing the amino acid methionine, binds to an AUG codon on the mRNA and recruits the large ribosomal subunit. The ribosome contains 3 RNA binding sites designated A, P and E. The A site binds an amino acyl tRNA, the P site binds a peptidyl tRNA and the E site binds a free tRNA before it exits the ribosome. Protein synthesis begins at a start codon AUG near the 5' end of the mRNA. mRNA binds to the P site of the ribosome first. The ribosome is able to identify the start codon by the use of the Shine-Delgarno sequence of the mRNA in prokaryotes and Kozak box in eukaryotes. Although catalysis of the peptide bond involves the C2 hydroxyl of RNase P site, adenosine in a protein shuttle mechanism, other steps in protein synthesis are caused by changes in protein conformations. Since the catalytic core is made of RNA, ribosomes are classified as ribozymes, and it is thought that they might be the remnants of the RNA world. Both ribosomal subunits, small and large, assemble at the start codon. The ribosome uses tRNA that matches the current codon on the mRNA to append an amino acid to the polypeptide chain. This is done for each triplet on the mRNA, while the ribosome moves towards the 3' end of the mRNA. Usually in bacterial cells, several ribosomes are working parallel on a single mRNA, forming what is called a polyribosome or polysome.